all this and for providing now this opportunity to talk about delivery and targeting mechanisms of biological macromolecules. So this, I'll probably take this here. So this is a proposal that, or a joint proposal from Sanofi, from GlaxoSmithKline, from AstraZeneca and Novo Nordisk. And it's actually interesting how it got together, because two of the companies, Glaxo and ourselves from Sanofi, we provided independently from each other a very similar proposal on delivery of oligonucleotides and delivery of therapeutic proteins. And then AstraZeneca and Novo Nordisk joined. Actually, at the beginning was also Genzyme part of it. And in the meantime, as you probably all know, Genzyme is part of the Sanofi group. Now, all this is basically a sign or the result of the desire of these companies to strengthen their portfolio with biologically based therapeutics. And there's a clear shift now in all the pharmaceutical companies to move away from, or the desire to move away, at least partly away from small molecules to biological molecules like therapeutic antibodies and also oligonucleotides. And this is really the result of this change in the industry, because all these biological macromolecules have a common problem, as you all know, and that's the delivery to the target cells and delivery into the cells. And this desire actually to move into biologics is the response to what we have heard this morning already from in one of the presentations this morning, is the result of this innovation gap or productivity gap that we have in drug development, reflected by an exponential growth of the R&D spending. And in spite of this growth of R&D spending, there's a decrease actually in the output, a decrease in new chemical entities that have been approved by the FDA, triple increase, but decrease of the output. And thousands of molecules are being worked on, and only a few actually make it then later on into the clinic and later on to the patient. So there's a huge attrition rate. And in spite of all these huge investments that have been made during recent years, especially into new technologies like genomics or proteomics, high-throughput screening, and so on, all in spite of all these huge investments, not much is coming out at the end. And this has partly to do with the therapeutic modalities that are being used in these various projects. So there's now a clear desire to shift away or to shift into new therapeutic modalities, including biologics. And there's another reason for it, because there's less generic competition, because it's not so easy to copy biologics like it is with small molecules. And another thing is actually here indicated here, there's a limited drug target space. In spite of knowing now the whole genome, all the current compounds in development are still directed only to a few hundred molecular targets. I think the number 10 years ago was about 500 of all the drugs that are directed against 500 genes, and this is still the same. So if there are opportunities to move into a new drug target space, and this is actually, as you will see later on, possible with microRNAs, for example, then this is going to be a huge opportunity. So this is clearly an environment ripe for change and new opportunities. And the new opportunities that we will talk about are the biologics. What we actually, as indicated from all this investment into genomics and new technologies, we have now a pretty good understanding of the disease mechanisms, of disease pathways, of biochemical pathways and biomarkers involved in diseases. We know which targets to drug, but we need to use the druggable targets, and we need to find the therapeutic modalities that actually get to the drug targets in the cell. And shift away from small molecules means to biologics like proteins, peptides, oligonucleotides, and what's also emerging is therapeutics with stem cells. What we want to focus on in our proposal for this consortium is to focus on proteins, peptides, and oligonucleotides as new therapeutic modalities, and move then these therapeutic modalities into translational medicine, into the clinic, and that's also what we can do very well, but where we really have problems is here in the middle to make this bridge between the target and the drug. So I'll give you a quick snapshot of some of the biologics that we want to work on. These are, of course, the therapeutic antibodies, which have been a huge success during recent years. Many new formats are being developed currently. Fragments of antibodies that are becoming smaller, and that's why it's then easier to get them to the targets or to the target tissues, to the target cells. So many new formats. Now the decision is, but the key problem is to bring these proteins still to the drug targets. There are even alternatives, non-antibody scaffolds being developed currently. There are many more formats that are also becoming smaller and therefore easier to handle, but it is here you can engineer basically antibody-like properties into new scaffolds. Then one thing that we want to point out is also what's really going to be, we believe, the future in therapies is oral delivery of peptides, of therapeutic peptides. Example is here insulin. And companies like Novo Nordisk and Sanofi do have interest in developing oral insulin formats. This is just an example. People have worked on this since decades already, actually, probably since the discovery of insulin even, to find a way for oral delivery of insulin. Nanospheres and nanocapsules, so many nanotechnologies have been developed and proposed to use those vehicles to bring insulin across this biological barrier in the intestine, this epithelial barrier, and we will talk about this more later on. There are different ways to get insulin through this barrier, but at the end, in spite of decades of research, and many biotech companies have tried that, there is no solution yet, no product yet that is really successful in the clinic. And insulin is just an example. There is glucagon-like protein one, which would be of interest, and many, many others, interferons, interleukins, and so on, many therapeutic peptides that where one would like to find a solution for oral delivery. And this might include in the future even larger proteins like antibodies or antibody fragments, and certainly also oligonucleotides, if this should be ever possible. Oligonucleotides, a lot of different formats have been tried, have been tested, a lot of oligonucleotides that can modulate the genome or modulate messenger RNAs, translation of messenger RNAs into proteins, or proteins directly intracellular or extracellular. Many different approaches, many different formats. The big hype was created through the discovery of RNAi, using siRNAs and microRNAs. But until now, there's only one oligonucleotide, antisense oligonucleotide on the market, and there's one aptamer on the market, and that's it.